Okay, everybody, can we can we get started? Oh, it's okay. All right. So today we're going to finish the fatty acid metabolism, the last lecture, the essential fatty acids, and after that we should have some time for review, right? For this Thursday, and we have exam 3. And for the coverage, I think I already mentioned in the email, all lipid metabolism, including today's lecture, right? For Thursday, and the format is the same, you know, the multiple choice and choose for selections. Again, and also 55 questions. And also I reserved room 153 for review session tomorrow from 1 to 2, right? From 1 to 2 o'clock, right? So you guys are welcome to come to the review session and just similar ask me question from either study guides or whatever you are not very clear want me to clarify, right? Okay, so essential fatty acids. What is essential fatty acid? What's the definition? So essential fatty acids are the fatty acids that are required in the diet. Why is it required in the diet? Because our body cannot synthesize them. And why we cannot synthesize them? Because we lack of enzymes, particularly the desaturases. So essential fatty acids mostly are polyunsaturated fatty acids, right? So it requires desaturases to make a double bonds. And we have some desaturases, like we have desaturase 5, desaturase 6, but we do not have desaturase 12 and desaturase 15 in our body. Right? So we cannot make some specific double bonds makes the specific polyunsaturated fatty acid. Right? So is that clear about the definition? The fatty acids that we cannot make in our body because of lacks the desaturases, particularly desaturase 12 and desaturase 15. And two classes of essential fatty acids, omega-3 and omega-6. Let's say the omega-6, the essential fatty acids in the omega-6 class or category is the linolenic acid, which has 18 carbons and 2 double bonds, right? Linoleic acid is omega-6 essential fatty acids. And this is the most abundant in the diet. And our body can synthesize arachidonic acid and arginic acid. So arachidonic acid, arginic acid are also omega-6 fatty acids, but they are not essential fatty acids because we are able to synthesize arachidonic acid and arginic acid from linoleic acid. So we must acquire linoleic acid from the diet to make arachidonic acid, arginic acid, right? And the other category is omega-3. The omega-3 essential fatty acids is alpha-linolenic acid, 18 carbons and 3 double bonds. And other fatty acids that belong to the omega-3 families are EPA and DHA. EPA has 20 carbons and 5 double bonds, and DHA is 22 carbon 6 double bonds, right? And again, in the omega-3 categories, alpha-linolenic acid is essential fatty acids. And EPA, DHA is not essential fatty acid because we can synthesize EPA and DHA from alpha-linolenic acid, right? So really, we only have 2 essential fatty acids that we need them from the diet. linoleic acid and alpha-linolenic acid from two different categories, right? Is that clear? Okay. So non-essential fatty acids, for example, other types of fatty acids, palmitate, oleate, other fatty acids that we know that usually are used for energy, right? We use palmitate, oleate for energy, right? We can oxidize these fatty acids and provide through beta oxidation, provide energy. And this non-essential fatty acids can also be stored in the body, right, as a triglyceride in adipose tissue, so as the stored energy. So in other words, the non-essential fatty acids, our body primarily use them for energy. But essential fatty acids is not metabolized for energy use in the body. They have a more important function in the body. They are the components of the cell membranes. Right? Cell membrane has many essential fatty acids, and they involve the other important biological process and functions. Right? So essential fatty acids, the function of essential fatty acids is not for energy. It's usually for the make cell membranes or serve as the signaling molecules to regulate biological process in the body, right? So the difference in terms of function between non-essential fatty acids and essential fatty acids. As structural fatty acids, I just wanted to remind you the numbering system, the omega numbering system. The first carbon from the methyl end is considered omega 1, right? Remember this numbering system. So how omega, 3 or 6 fatty acids, how we named this. So omega usually refers to the first double bond position, start from methyl end. For example, omega 3 fatty acids, you see the first double bond is located between carbon 3 and 4 from the methyl end, right? So this we usually call omega 3 fatty acids, and omega 6, the first double bond, start from carbon 6. So this is how we named omega 3 and omega 6 fatty acids, right? And depend on the location of the first double bonds in the fatty acids molecules. And he just gave you some idea, structure of the omega 3, omega 6 fatty acids, like alpha linoleic acid, which is omega 3 fatty acids, the first double bond at the 3 position. I have 3 double bonds, 18 carbons for alpha linoleic acid, and omega 6 fatty acids, linoleic acid, first double bond start at carbon 6 position, have 2 double bonds, 18 carbons. And arachidonic acid, EPA, and they have more double bonds, and just the first double bond is different, start a different carbon position. Mm-hmm. So let's talk about the more detailed about each category of fatty acids. Omega 6 polyunsaturated fatty acids family. We know that 3 most important fatty acids belong to omega 6 polyunsaturated fatty acid families, including linoleic acid, which is essential fatty acid, right? And arachidonic acid and argininic acid. So linoleic acid, the most abundant in the diet. The main sources of linoleic acid from the diet, including corn oil and sunflower oil, seed oil, and the safflower oil. And most abundant, again, this is the most abundant polyunsaturated fatty acids in the diet. And arachidonic acid, which has 20 carbon 4 double bonds, and can be synthesized in the body from the dietary linolenic acid, right? So arachidonic acid is not essential fatty acids. And the main substrate, so arachidonic acid is the main substrate for synthesis of eicosanoid. Eicosanoid is the very important so-called biomediate or the lipid metabolites that involve a lot of biological activities, right? For example, prostaglandins and leukotrienes, and these are the most important biomediators that are synthesized from arachidonic acid, right? And you guys know this biomediators that involved in the inflammation, right? The local inflammation, the prostaglandins and leukotrienes, very important. And these are the lipid mediators, right? And synthesized from arachidonic acid. And arachidonic acid usually is converted from arachidonic acid by elongation and can also be converted back to arachidonic acid. So these two fatty acids can be interconverted each other, right? And most important one is the arachidonic acid, right? It's the substrate for the synthesis of prostaglandins and leukotrienes, the mediators involved in the inflammation, local inflammation. And the alpha-3 polyunsaturated fatty acid families, and including, again, also major 3 fatty acids, alpha-linolenic acid, which is essential fatty acids, and most important source of the alpha-linolenic acid are the soybean oil, looks like probably low battery. Change battery. Okay. Oops. Okay. So, yeah, the most abundant, the major sources of a alpha-linolenic acid, soybean oil, and canola oil, and vegetation. And vegetable oil, I think. So the EPA, although we can synthesize EPA, right, DHA in the body, but they also very rich in the food, in the diet. For example, fish oil are the major sources of the EPA and the DHA. So EPA, DHA are the good fatty acids, polyunsaturated fatty acids, right? It can have a lot of studies showing they have a anti-inflammation, functional effect. So omega-3 fatty acids normally considered a good fatty acid, and omega-6 it depends, because they are the major sources of inflammatory biomediators, right? So. Okay, now let's talk about the metabolism of essential fatty acids, how essential fatty acids in the body are metabolized. So body usually utilize the dietary linoleic acid or alpha-linolenic acid to synthesize all other forms of alpha-3 and omega-3 and omega-6 fatty acids, right? We already talked about. And synthesis of omega-3 and 6 fatty acids usually being complete by the same reactions or pathways involved in fatty acids elongation, desaturations, and literate conversion, right? But these two categories, omega-3 and 6 fatty acids, cannot be interconverted, right? So they synthesize separate, but they cannot be interconverted. Although they use the same pathway to synthesize in the body. Okay, here is the sort of pathways, how omega-6 and omega-3 fatty acids be synthesized from essential fatty acids. Alpha-linolenic is essential, again, fatty acids in the omega-6 family, and alpha-linolenic acid is the essential fatty acids in the omega-3 fatty acid family, right? So involve the two enzymes, delta-5 and delta-6 desaturase, right? So we have a delta-5 and delta-6 desaturase in our body, so our body able to synthesize the these other families of omega-3 and omega-6 fatty acids from essential fatty acids. And I'm not going to go into detail, but first of all, desaturate to make a double bonds, and then elongate to extend the carbon chain, and then do another desaturating at another double bonds, and elongate, extend carbon chain, and third double bonds, right? And then the Leach conversion means that the you synthesize longer carbons, fatty acids, you can be remove the carbons and convert it back to shorter fatty acids, shorter chain fatty acids, right? So just try to remember the two categories, the omega-3 and omega-6 fatty acids can be synthesized from essential fatty acids and through the same pathways, right? Elongation and desaturase. And remember, we have delta-5 and delta-6 desaturase in our body, right? So here, just a little bit detailed, the process of the synthesis omega-6 fatty acids from essential fatty acids like linoleic acid as a substrate, right? And you do the desaturation and utilize NAD to add the double bonds. And then you extend carbons, so elongase enzyme, right, to catalyze the extension of the carbons, and try to notice that the carbon actually extended between the carboxyl carbon and the double bonds that close to the carboxyl group, right? It's not added from the methyl end, right? It's adding between this, double bond and the carboxyl group. And then the second desaturation, right? You add the double bonds between this carbon. So again, the substrate is a linoleic acid and requires the marino-CoA as the carbon donors, like we talked about earlier, right? The fatty acid synthesis pathway, the same similar pathway. You're adding marino-CoA to extend 2 carbon at a time, and need the NADH or NADPH as the donor of the hydrogen, right? And the enzyme that involved this synthesis saturase, delta-5, delta-6, and elongase, right? And the product, here we talk about the arachidonic acid for omega-6 fatty acids families. So I already mentioned the function of the omega-6 is primarily used for synthesis of eicosanoids, including prostaglandins and leukotriene and lipoxygens and other lipid mediators, right? And particularly, prostaglandins and leukotrienes are very important. the regulators of biological activity, particularly involved in the inflammation, local inflammation. So when inflammation is response increase, then the secretion or synthesis of prostaglandin and leukotrienes increases, so mediate the inflammatory response, right? So this is sort of the main function of the omega-6 fatty acids, arachidonic acid. And here we also wanted to mention the other families of omega-6 fatty acids like linoleic acid. Linoleic acid, in addition to serving as a substrate for arachidonic acid synthesis, right? And it's also important, a component of the sphingolipids, and this is also important for sort of making skin water barrier, to protect kind of dryness of the skin. And the arachidonic acid I already talked about is the primary substrate of eicosanoid and prostaglandins, right? And this are the important component of phospholipids and signaling transduction to regulate, the biological activities. And other things about omega-6 fatty acids, some of the product of omega-6 fatty acids like eicosanoids, prostaglandins, they are important ligands for PPAR gamma. PPAR gamma is a key transcription factor that regulates the lipid metabolism, right? So, but it's okay if you don't, remember this. Just try to let you know how omega-6 fatty acids are important in regulate, biologic activities, the lipid metabolism. Okay, so here is the pathway how this biomediators, prostaglandins, leukotrienes are synthesized from arachidonic acid. So 3 pathways involve the 3 key enzymes that regulate the synthesis of biomediators from arachidonic acid, right? Like cyclooxygenase, lipoxygenase, and cyclone P450, and these 3 enzymes, right, that's very important, regulate biosynthesis of prostaglandin and leukotrienes from arachidonic acid. And again, I mentioned this arachidonic acid, leukotriene, all these lipid mediators are involved in the local inflammatory response. Now let's talk about omega-3 fatty acids. So alpha-linoleic acid is essential. Omega-3 fatty acids, and they usually used for synthesis of EPA and DHA in the body. And not very clear about the unique function of alpha-linoleic acid. But it's important for the synthesis of EPA and DHA. And EPA, DHA are the important omega-3 family of fatty acids, polyunsaturated fatty acids. They have anti-inflammatory functions, right? And EPA, DHA is also important substrate for the synthesis of resolvins and protectins. Resolving protectins also are the lipid mediators, right? The lipid metabolites from DHA and EPA. And these lipid mediators or metabolites are very important, play a role as anti-inflammatory, right? Anti-inflammation. And you probably also notice that this synthesis of resolving and protectins from EPA and DHA also utilize the same 3 pathways, same 3 same enzymes, right? And as the omega-6, remember arachidonic acid, right? Also utilize the same enzyme, same pathway to synthesize the pro-inflammatory mediators. So here just try to explain why EPA or DHA has an anti-inflammatory effect. Remember that these two, EPA or DHA and arachidonic acid, utilize the same enzymes to synthesize either omega-6 eicosanoids like prostaglandins, leukotrienes, right? Or for omega-3 families and utilize omega-3 eicosanoids, which like resolvins and protactins, right? They are actually anti-inflammatory. So if we if we consume a lot of EPA or DHAs, right, then we can compete the enzymes, 3 enzymes, with arachidonic acid for the synthesis of omega-3 eicosanoids, which is a good anti-inflammatory lipid metabolites, right? Does that make sense? So that's one of the mechanism. You know, we try to eat more EPA or DHA to compete with the arachidonic acid, 3 omega-6 fatty acids for metabolizing enzymes and for synthesis of good mediators, biomediators, right? So this is sort of the one mechanism. And in terms of function of omega-3 fatty acids, it's a smaller molecules and less bioactivities, but it's very important to inhibit omega-3 eicosanoid production, right? If we have more omega-3 EPA or DHA, we can inhibit pro-inflammatory or bad eicosanoid production, right? So omega-3 eicosanoid usually have an anti-inflammatory effect and can increase fatty acids oxidation and decrease triglyceride levels in the plasma, and it also can inhibit the triglyceride synthesis in the liver, right? It's just summarize, you know, the research findings and related to the omega-3 fatty acid effect, right? And the mechanism is still not very clear. And DHA function is DHA mostly present in the phospholipids of cell membrane, right? It's important for the constituting the cell membrane, and it's also rich in the retinol and brain. It's, you know, sometimes it's important for the visual actions, you know, cognitive actions, and okay. So yeah, I'm not going to talk too much about this. Probably you know, guys don't remember. Just in general, I mean, omega-3 fatty acids are the good fatty acids, right? It has anti-inflammatory effect, and one of the mechanisms is just to compete for the metabolized enzyme with the omega-6 fatty acids, so can reduce the omega-6 eicosanoids or metabolites. Yeah, this is something more specific sort of molecular metabolism, molecular mechanism, just how you know this omega-6 or 3 fatty acids regulates the metabolism. Basically, the control these 3 key transcription factors: the PPAR gamma, XLXR, and SIEBP. You remember this one, right? So regulates the activity of this enzyme and regulates the lipids, the glucose metabolism. right? Okay, so just want to summarize the essential fatty acids, what you guys need to know. First of all, which fatty acids are the essential fatty acids? Only two, right? Alpha linolenic acid, alpha linolenic acid from each category, right? Omega-3 and omega-6 fatty acids. And the reason why they are essential because they cannot be synthesized in the body and it has to obtain from the diet. And the reason why we cannot synthesize these two essential fatty acids. Why we can't? Yeah, we lack desaturates, right? Which desaturates? 12 and 16, right? And which desaturates that we have in the body? 5 and 6, okay. What's that? 15, yeah, I'm sorry. Did I say 16? Sorry. 12 and 15, right? Sorry. So human bodies, yeah, I've already talked about this, and and the synthesis of the other families of fatty acids, like arachidonic acid, EPA, from essential fatty acids, it goes through the same pathway, right? Elongation, desaturating, and substrate are the essential fatty acids in each categories, right? And again, the two carbon donors, malonyl-CoA, right, also requires the NAD, H and NADPH. And also you wanted to know that the arachidonic acid for omega-3 fatty acids usually are used for synthesis of pro-inflammatory eicosanoid, right? Omega-6 is a pro-inflammatory, including prostaglandins and leukotrienes, and omega-3 fatty acids, DHA and EPA are good fatty acids, and they synthesize the resolving and protecting, right? It has an anti-inflammatory role. And these two categories of fatty acids can compete each other for the metabolized enzyme, right? Yeah, I guess that's all you guys need to know, right, for essential fatty acids. Now do you guys want to take a break and then we come back to the review session, right? Yeah, 10 minutes break. right. So let's start the review session. Okay, so questions. Which part do you guys want me to clarify? Or what kind of question you want me to address? Yeah.
Can you go over the increase and decrease in the cholesterol pool?
Cholesterol pool, you mean the cellular?
Yeah.
Okay. Can you tell me the unit? 3, 4? Okay. What's that? Probably 5, 6, right?
And then you covered some of it in 7 as well, the cholesterol, cellular stores.
Okay. Oh, I guess, yeah. Sorry. Seven? No, same. No? Shoot. I don't know why I can't find it. This is a 7, 5, 6. Don't see it. Oh, here, right? Sorry. Okay, this slide that you want to. Okay, so this we talk about the intracellular cholesterol homeostasis, which is important, right, related to atherosclerosis development. So you try to remember the pathway that, the intracellular pool of free cholesterol, right, and pathways that contribute or adding cholesterol to the cell pool, or you remove the cholesterol from the cellular pool. So these two pathways, one adding and one remove, you need to be balanced. So you try to remember the different pathways that can contribute or adding the cholesterol to cholesterol pool, which including uptake, right? Uptake from the circulation, from LDL receptor mediated. So you're adding, you take up cholesterol from circulation into the cells and adding to the pool, right? And also synthesis, cholesterol synthesis, right? Cholesterol can be synthesized in the cells and also adding the cholesterol to the pool to making high levels of cholesterol inside of cells, right? And the third pathway is that the cholesterol and free cholesterol ester is also kind of balanced in the cells. So when this pathway is increased, you have more cholesterol ester being break down to free cholesterol, while you also increase the free cholesterol level in the cells, right? Just thinking which pathway can regulate the cell cholesterol levels, right? And for the reducing cell cholesterol pathways, for example, we remove cholesterol from cells and adding to the HDL. HDL is a good cholesterol, right? The lipoprotein can take away cholesterol from the peripheral cells to the liver, right, for disposal. So this is the most important pathway that can reduce cellular cholesterol by taking the free cholesterol and load it to the beta HDL, right, and do so-called reversal cholesterol transport pathway and deliver to the liver. And again, try to remember ABCA1 is an important transporter, transport free cholesterol from the cell to the pre-beta HDL, right? And the second pathway is for synthesis. Free cholesterol can also be utilized for synthesis of steroid in the cells, right? If you utilize some free cholesterol, you also reduce the cellular cholesterol levels. And the third pathway is to re-acidify and to cholesterol esters, right? You can also reduce the free cholesterol levels. Does that make sense? So you just try to thinking, which pathways can increase cholesterol level in the cells and which pathways can remove cholesterol from the cell and reduce the cholesterol inside of cells. And use, yes?
How are those LDL receptors mainly regulated?
What's that?
The LDL receptors, how are those mostly regulated?
Oh, the cell receptors? Yes. So this receptor is the most important, right? They control your uptake. So anything that have a defects in the LDL receptor itself or the apoprotein that can be recognized by LDL receptors also important, right? To control the rate of the uptake of LDL, right? So you guys remember which apoproteins that are important for LDL receptor uptake, LDL? ApoE and ApoB100, right? So try to remember these two. So if, for example, if ApoE deficiency would happen to the blood cholesterol levels will be increased, right? Because it cannot be taken up by the cells. And also try to remember LDL receptor most abundant in the liver, right? So means that uptake mostly occur in the liver. So if liver cannot pick up a lot of LDL, then you raise the LDL level in the circulation, and LDL will be picked up by the scavenging receptors in the peripheral tissues, right, including macrophages. So if macrophages pick up a lot of cholesterol and in the circulation and then become the firm cells and develop atherosclerosis, right? Any other questions? Yeah.
Review the beta oxidation.
Beta oxidation, okay.
It's in
3 and 4, all right. Okay, beta oxidation. So let's just start with like fatty acid oxidation. So fatty acid oxidation means that we break down the fatty acids to the small molecules, right? Which the end product, which molecules are end products of fatty acid oxidation? Acetyl-CoA, right? And so the first important reactions that the activation of fatty acids, right, by called a fatty acyl-CoA synthetase. And this reaction is actually common for both lipogenesis and the fatty acid oxidation, remember? And then this reaction occurs in the cytosol, and once it's activated to fatty acyl-CoA, and fatty acyl-CoA need to go into the mitochondria, right? And so the transporter of fatty acyl-CoA from cytosol into the mitochondria requires as a key enzymes on the membrane of mitochondria. Remember which enzymes? CPT1, right? CPT1 is the enzyme that control reaction, and this beta oxidation goes also cycles. Each cycle you just remove the 2 carbon units as the acetyl-CoA, right? And so you try to remember, for example, palmitate 16 carbon fatty acids, how many acetyl-CoA being generated, and go how many cycles of beta oxidation, right? So this occurs in mitochondria, and this is the key regulators of beta oxidation, right? Yeah.
So does CPT not need energy for ATP?
No. Right?
What's the function of
Fatty acyl synthetase? Sorry, what?
fatty acyl the enzyme?
Right. What's your question?
What's the function of it in terms of beta oxidation?
Oh, and this is just the first reaction, right? Before fatty acids being going into the mitochondria need to be activated to the fatty acyl CoA, just adding the CoA group to the fatty acids, right?
Also, are fatty acids or on-chain fatty acids glucogenic?
The-
Are on-chain fatty acids glucogenic?
Out-chain fatty acids glucogenic, yes, because the last one of the end product is the proline-CoA, right? Proline-CoA is the 3 carbons. So 3 carbons can be used as the glucose neogenesis, right? Okay. Other questions? Sorry?
For the beta oxidation?
Yeah.
Oh.
More questions about beta oxidation?
Do we have to know like all of the steps?
No, don't need to do the details like which enzyme or, no, just the general, right? The process, each cycles and remove the one acetyl-CoA, right? 2 carbons, it's acetyl-CoA.
So that each cycle, like just the basic like reactions right here?
Yeah. And, right. Yeah, and you know how many FAD or NADH produce, and you probably try to calculate the ATPs, just, to be familiar with the process. What's that?
What are the hormones that play a role in lipolysis and exercise?
What hormones plays a role in lipolysis exercise? Catecholamines, like epinephrine, right? Norepinephrine. Actually, insulin also involved, because insulin basically suppresses lipolysis, right? But during exercise, because a lot of glucose being taken up by the muscle, so glucose level is also decreased. So that would decrease insulin secretion. So insulin levels decrease, but catecholamine levels increase. So they combine, work together to increase lipolysis. Yes?
So for this slide,
Yeah, I think it's just to know, you don't have to remember exactly each reaction, but how many NAD or FAD being produced, right? And you see the one cycle that you remove one, the acetyl-CoA, right? Something like this. What's that? Enzymes? No. Yeah.
Can you go over the differences in HSL and LPL?
Okay. So difference between HSL and LPL. Let's thinking about in the adipose tissue, right? So first of all, which physiological conditions that makes HSL or LDL. Right? First, the LPL usually is induced, right, or is increased by insulin during the fat condition in the adipose tissue, right? And HSL basically is increased during the fasting. So there's the difference in the, the stimulation or regulation in different physiological state. And secondly, the location is also different. So HSL is localized inside of cells, right? The adipocytes, and LPL is on the considered kind of circulation, but in the capillary wall, right? Close to the endothelial cells. It's not inside of cells, outside of cells, we'll say, right? So this is the second difference, and the third one is the substrates. They both catalyze the same reaction, like hydrolyze triglyceride to release fatty acids and glycerol, but the substrate location is different. For example, HSL, hydrolyzed triglyceride inside of cells, right? The adipose cells. And LPL, hydrolyzed triglyceride in lipoprotein, remember? Chymicron and VLDL, right? So I think that these are the major three aspects of these two enzymes different, right? Any other question? Just try to understand, right? Stuff, and then that will help you remember. Try to remember key things and then understand the whole pictures, right? Oh yes. Mono isoglycerol?
Yeah, the monoacylglycerol formation. There's
Oh. Just to try to remember the triglyceride synthesis, right? You're adding 3 fatty acids to the glycerol backbone. And the first SN1 is usually saturated, right? And SN2, the unsaturated fatty acid, something like this. And then the last step, the enzyme that DGAP, right? The enzyme to catalyze the last fatty acid has been added to the SN3 position, is the key enzyme that control the whole triglyceride synthesis rate, right? Yeah.
So I know there's the same enzyme in ketone body synthesis and cholesterol synthesis. I know it says synthetase conase, or HMG conase, synthetase conase.
Mm-hmm.
Ketone body synthesis, and I thought it was HMG conase reductase, cholesterol synthesis.
No, HMG-CoA reductase is a key enzyme for cholesterol synthesis, right? And HMG-CoA synthase is enzyme that catalyze the reaction that are common for both cholesterol synthesis and ketogenesis, right? Right? Yeah.
Can you go
VLDL. Okay. VLDL basically is made from liver, right? And it's actually different. The difference is the the fatty acids that being loaded on to the VLDL as a triglyceride, the sources of fatty acids different between fat and the fasting condition. So VLDL can be made from the liver in both fat and fasting condition. In the fat condition, the triglyceride, the fatty acids in the triglyceride, primarily from diet, right? And during the fasting, the fatty acids in triglyceride in VLDL from the fatty acid from the lipolysis of adipose tissue, right? And also as the condition that the diabetic condition, we have a lot of fatty acids in the circulation, and so the triglyceride is also from the circulation. What? No, the diabetic condition. So diabetic condition, the lipolysis rate is increased. So you diabetic patient actually release a lot of fatty acids from adipose tissue. So that can also be packed in the VLDL. And VLDL basically is taken up by the adipose tissue, right, in the fat condition, because this fatty acids initially from diet, either from the chylomicron remnant or from de novo lipogenesis. Remember this, right? So these fatty acids need to be transported to the final destination in adipose tissue. So adipose tissue basically picks up the fatty acids or triglyceride from VLDL in the fat condition, right? And in the fasting condition, that VLDL, because I talk about the VLDL synthesized that transported to the muscle, right? The fatty acid initially from adipose tissue, but they need to be utilized by the skeletal muscle. So the destination is different in the muscle, right? Yeah.
Remember how the SIBP controls HTG, right?
SIBP?
The transcription factor.
Oh, the transcription factor, SIBP. You just need to know how cholesterol regulates SIBP activities, right? So SIBP basically is increase the transcription of HMG-CoA reductase, right? So and cholesterol levels, if it's increased and decreased cholesterol synthesis, right? So decrease the HMG-CoA reductase, and basically is decrease the SIBP activity, right?
Cholesterol does?
Yeah, cholesterol levels increase and decrease SIBP, decrease HMG-CoA reductase, right? And then inhibits cholesterol. Yeah, the same regulate mechanism. Oh, SRE is like the DNA sequence that localizes the upstream of the HMG-CoA coding sequence. So the SIBP binds this DNA sequence and triggers, the gene transcription of HMG-CoA reductase. But you don't need to know that details, right? Right? Yes.
For the lipid metabolism 7, do you know the specifics for each clinical case?
Yeah, I think that's actually quite important, Oh, really? sorry. So I think this is very important, right, to help you to understand all the material that we learned from the lipid metabolism. So we try to understand the cases, each case is how they, you know, I mean, how the what do I try to say, they work for the atherosclerosis development, you know, something like this. So in general, I think try to remember, again, I talked about earlier, just liver is a major place that pick up LDL. If something's wrong with either apoprotein E or apoprotein 100, then LDL cannot be picked up by the liver, then more LDL will go to the peripheral tissue, picked up by peripheral tissue through the scavenging receptor, right? And increase the cellular cholesterol level, and you know, and promote the atherosclerosis. And ABCA1 receptor transporter, right, is also one of the cases I remember. Is, you know, remember that where's ABCA1 located in terms of peripheral cell surface, right? What is the function of the ABCA1 is to pick up to transport the free cholesterol from the cell to the beta HDL for reverse cholesterol transport, right, to the liver. So ABCA1 is a good thing, right, to remove the cellular cholesterol. And also the other enzyme, you know, the reverse transport cholesterol, you try to remember the whole process, which start first and, you know, and to what, and the several steps occur in the plasma, involve CPT, CEPT, the transporter, right, the lipid exchange, and also the reacidification, free cholesterol to cholesterol. So we try to remember several protein names and what are their functions in the cholesterol reverse cholesterol transport process. Right?
Can you go into more in depth with that cycle?
Remember you know, ABC1 transfer free cholesterol, right, from the peripheral cells to loaded free cholesterol to the pre-beta HDL, right? And beta HDL also receive the phospholipids, right? Both free cholesterol and phospholipids, and then become sort of discodal HDL, and then released into the circulation. And in circulation, I think the reacidification of the free cholesterol to cholesterol ester, that reaction requires the LACT, right? Sorry, I can't remember exactly. The enzyme to catalyze the acidification of the free cholesterol to cholesterol esters, right? And then there are also the lipid exchange between HDL and VLDL, IDL, and LDL, right? And the enzyme that responsible for the or protein responsible for the transfer of lipids, right? Remember CEPT, right? This protein, and then eventually become alpha HDL, right? Alpha HDL then be picked up by the liver. So you deliver the cholesterol from peripheral tissue to the liver, and for synthesis of bile acid or secretion, and or some of the cholesterol also being used for synthesis of steroids, right, in the liver, right? And also, you guys probably wanted to remember the different, you know, the lipoproteins, their composition, and apoproteins, which apoproteins are unique to which lipoproteins, right? And where they made from, and you know, where they are made or synthesized, and what is the function of each lipoproteins or apoproteins, right? So you probably wanted to remember the ApoE, Apo 100, ApoC2, right? We just have a test question. ApoC2 is the required for LPL activities, right? And so if we don't have enough ApoC2, that we cannot activate LPL enzyme activities, right? So then cannot be hydrolyzed triglyceride in the chylomicron and VLD, also eventually will raise the triglyceride level in the circulation, right? So this, right? Yeah.
One more thing about the cycle. When the alpha HDL goes to the liver, then hepatic lipase breaks it back down to beta HDL.
Right. So the the hepatic lipase is in the liver, so it's responsible for the hydrolyzed triglyceride in that being picked up by the liver, right? Either chylomicron or alpha HDL. Right?
So what does the liver take to make the bile acid? It takes
The cholesterol esters, yes. Yes? That's a good question. So HDL is also need to be taken up into the liver, and then they just hydrolyze the triglyceride inside of the HDL, right? And also remove the cholesterol ester. Right? More questions? So try to focusing on the class notes and make sure that you understand all, materials and stuff that we discussed in the lectures. And then also try to, combine the study guide, right? Study guide. Maybe some questions, some information we missed in the study guide, like you just mentioned, the 3 cases, this kind of things is very important actually. So, yeah. Oh, the 3 cases, the like mutations in ApoE proteins, or I think it's 7, right? The clinical relevance. So we talk about 3 cases. Yeah.
So after essential fatty acid lecture today, should we just look at the summary of your points instead of like the...
Essential fatty acids, you mean?
Yeah, sorry.
Oh, yeah. So essential fatty acids, I think we just need to know which fatty acids are essential fatty acids, why essential, and the some of the function of the omega-3 fatty acids. Right? So I guess probably the lipoprotein metabolism is the most confusing stuff, right? Right? And if you guys have more questions when you review, material, and you can come again tomorrow. right? 1 to 2 o'clock.
Just a little better understand this question, so can you read that answer?
So what happens to the rate of ketogenesis when serum fatty acids level is high? So you said increase, right? Yeah, and you mean this one, right?
Yeah.
Why? Okay. So
Type 1 diabetes is because of the lack of insulin, right? So what insulin does is to transport glucose from circulation into the cells. So if we don't have insulin, then this process will be decreased, right? So we can transport the glucose into the cells. So make it kind of a two situations. And in the circulation, you have more glucose being remain, right, in circulation, but actually you have a much lower glucose because you can't get glucose into the cells. So for cells, they are actually lack of glucose, right? So they kind of go energy crisis. So if cells do not have enough glucose for energy, then they have to use more fatty acids, right? Need to use more fatty acids. So that will stimulate the fatty acids mobilization and increase circulating fatty acids levels, and fatty acids being picked up by the liver and oxidize and produce a lot of acetyl-CoA, right? And then promotes the ketogenesis in the liver.
Okay.
So there's something.
So I got that pretty right. Insulin
Mhm.
regulator, TCA, so it's absent, rise and increased beta oxidation. Is that correct?
Yeah, let me see what it is. I don't know where to be. Two options. Yeah, maybe she thought you did not mention, that the glucose utilizations role that is the main drivers for the increased fatty acids
So
utilization.
insulin does not regulate free fatty acid metabolism directly.
No, insulin. Yeah, insulin is
Does insulin regulate lipolysis?
right. So yeah, so if you mention the I don't know what it is. So, okay. Numbers increase. Okay. Yeah, mostly right. I think it was this part.
It's just that
Insulin
insulin does not
insulin.
directly
Right.
stimulate beta oxidation, but it stimulates lipolysis.
Insulin basically suppresses lipolysis, right? So, and if insulin is to decrease, then you have an increased lipolysis, right? So you have increased fatty acids release, something like this.
And the
Did you
increase in
mention this?
fatty acid-
Did you mention this?
Said free fatty acid levels rise and beta oxidation increases,
Right.
which forms ketones.
Yeah.
So that's right?
And right, but you probably didn't mention, why fatty acid levels
Why
rise.
beta? Because beta
Right.
oxidation only increases because of lipolysis.
Right.
Right. Okay.
Right.
Thank you.
Oh, yeah, you're welcome.
Hi.
Hi, is the final
Final, yes, actually 70% the material from the rest of the,
70% is from the new material?
the new materials, yeah, and 30% right.
Okay,
So
yeah, that's not too bad.
not too bad, right?
Okay, thank you.
Your quiz? Okay. Yeah. Oh.
Yeah.
So I'm just a little confused by this, what this is saying exactly.
Mhm.
That type 1 diabetes
Right.
causes hyperlipidemia. Is that not true?
Um. think you. Yeah, I think mostly you're right. Okay. What point? How many points you get?
I
One.
got 2 out of 3.
Ah,
I think I got
level
marked
three.
down 1 off of that.
Increases and increases. Okay. Okay. Yeah, I can give you that on this point. Can I keep this?
Yeah. And then,
What about
I don't know,
this
is it
one?
too late to talk about quiz 3?
Quiz 3?
Yeah. No, that was.
It's also your. Okay.
I was just wondering,
What's the question?
here I lost a point, because I know Malone Alcoa donates like the 2 that build
Right.
on it.
Mhm.
And I did say that. I don't know, I just wanted to be thorough and make sure that it's like Acetyl CoA donates the first 2, and
Mhm.
as it's elongated Malonal. And I know someone else was looking at her answer and she said Malonal CoA donates the 2.
Hmm. I see. Okay. Yeah.
Thank you so much.
Yeah. Okay. Oh, okay.
Next Tuesday,
Mhm.
so I've been in touch with a professional, a dietetic, in the field of food science nutrition. And the only time that she was able to meet was next Tuesday during this conference.
Okay.
I know what was on Monday, and
Right.
we have an A class Friday.
Right.
I have been working on the homework, and I can get that in
Mhm.
beforehand.
Mhm.
I was wondering if there was any way I could possibly make
Okay.
that A class Friday.
Yeah, why don't you send me email on that day, then will send you a question, and you can email me back the answer.
Thank you so much.
Okay.
So I was out of something after I went back up there. So
Yeah.
you said that I could get that half point back, and then this one that she thought I missed is on the back. So I mean,
Oh,
I'm pretty
okay.
sure I got that right.
Mhm.
But it said I missed 2.5, but that just adds up to 1.5. So with those points, I would get 15 or 15.
Oh, what's that? I'm kind of confused.
Yeah, because
Um,
I was just like so confident after this quiz, and I thought 12.5. I'm like, how did that?
So this will be 1 point, right? And
1
this will
point,
be
but I wrote it on the back, and she didn't see it.
right.
So I know I
So you will
answered
get
that
1
right.
point and point 5, right? So 1.5, 1.5.
Yeah, but
14.
I only missed 1.5, so she did the math wrong.
Ah, really?
Yeah,
Oh.
because there's no other point off.
I see. Oh, okay, okay. Oh yeah, I mean if that is the case, yeah.
Okay, so you remember when I came to your office and I was all like hopeless and crushed, and I'm doing so much better. But I have one question
Mhm.
that just a question. I was wondering, so you know we get to drop our lowest grade. Is there any chance we get to drop our 2 lowest quiz gradings?
For quiz,
Because it
probably
would definitely help
no.
a lot.
Probably not for this semester, I guess, because we only have, actually we already reduced the quiz. Last semester we have 8, and now we have a
Maybe we
6.
could add 1 extra and then drop 2.
Well, you know, I usually would do the curve at the end, you know, for the final grade. I usually would do this. So I probably wouldn't worry too much at this point.
I'm gonna wait until I see my grade. It's
Mm.
really borderline. Obviously, this exam will add.
Yeah. I will have to see, see how the whole class performance, and then maybe, I'm not sure yet, maybe we'll do sort of like a bonus question, like extra 3 points or something, that if you do it, then you can get extra points. So, yeah. My goal
goal is to make everybody happy. Yeah. Yeah, you're welcome.